Periodically, we take great pride in hearing from those researchers here on campus that we have helped support through Kennedy Center research grants. And today we have one of those opportunities as we welcome Dr. Keith Crandall from the zoology department here at BYU. Dr. Crandall earned his BA from Kalamazoo College and his master's and PhD degrees from Washington University in St. Louis. He finished his PhD in 1993, after which he served as a postdoctoral fellow at the University of Texas for 2 years before joining the BYU faculty in 1996, where he's currently an assistant professor in the Department of Zoology and concurrently an assistant curator at the Bean Museum here on campus. Dr. Crandall is involved professionally in a number of ways, reviewing journal articles, making presentations, and conducting research in the area of biodiversity. And that is the topic upon which we have asked him to present today. So please join me in welcoming Dr. Keith Crandall for a presentation on biodiversity studies at BYU.
Well, thanks very much. It's a pleasure to be here. You guys, I understand, have had a lot of political sorts of talks, and this is going to be something completely different. We're going to talk about biology today. We're going to talk about biodiversity and why it's important and why you should be interested in it, and how much fun it is to study and how much it is being studied here on campus by professors at BYU through international collaborations around the world, and how we incorporate undergraduates in our research efforts. So at the end, I'm going to encourage you all to get some research experience. You don't have to be biology majors to sign up for some of these fun activities. So the advertised title of the talk is Biodiversity Studies in the Amazon. The actual title of the talk is Biodiversity Studies at BYU. We're going to talk about the Amazon, but we're going to branch out as well and travel around the world a bit, because that's the fun part of biology, being able to travel around the world. So the first question is, what is biodiversity? Why are we interested in this stuff? And this is a definition that comes from E.O. Wilson, who's one of the foremost conservation biologists in the world today at Harvard University. And he defines it as all the hereditarily based variation at all levels of organization, from the genes within a single local population or species to the species composing all or part of a local community, and finally to the communities themselves that compose the living parts of the multifarious ecosystems of the world. So biodiversity studies, not only it's not simply species counts, but it's more broad than that. It does include species counts, but it also looks at the genes within those species and the diversity, the genetic diversity that's housed within those species, and at the species as they come together in diverse communities and the diversity of communities. So it's an all-encompassing topic, and it's a really hard idea to get your head around, and it's a really difficult topic to study. So in biodiversity studies, we're interested in a number of things. Here I've listed three primary emphases that we're interested in biodiversity studies. First is to discover and describe and inventory global species diversity, and this is a species count. I'll show you in a minute. It's impressive how little we know about the organismal diversity in the world today. Secondly, to develop a predictive classification system that reflects the history of life. So not only do we want to describe each and every species that exists in the world, but we also want to know their relationships to one another and use that relationships to classify the organisms so that classification becomes much more powerful as a tool in using biodiversity information for other purposes. And I'll get into that in a minute. And then finally, we use biodiversity studies to organize this information in a retrievable form that meets the needs of science and society. And this means that we do a lot of databasing, a lot of database management. There are a lot of biologists with significant computer skills in figuring out how you can develop good databases with good retrieval systems, and then make that information accessible to the public. And we're hot and heavy into this at the Bee Museum here on campus. We're in the process of computerizing all the collections at the museum, and they're significant collections. The insects alone, we have over 1 million specimens. One of the best collections of Great Basin insects in the world. So, and that's just the tip of the iceberg over there at the museum. So we're in the process, as you can imagine, cataloging 1 million specimens on a computer system and then uploading that to the web so that it's searchable and accessible. So if you want to know what stoneflies occur on the Provo River so you can design your tie appropriately when you go fishing next week, you can access that information. If you want information about the emergence of particular flies on particular rivers at particular times, you want to be able to do a sophisticated search, right? So if you want to go up to Deer Creek and go fishing or one of the nice trout streams and a particular time of year, you want to know what kind of fly you need to tie, right? So you have to have these accessible databases, right? We don't just do them for fly fishermen, although that is a good reason. So one of the motivations for doing these biodiversity studies is the fact that we really have no good idea how many species there are. And here's just some examples. In viruses, there are over 5,000 species of virus described to date, and it's estimated there are over 500,000 species. For bacteria, there are 4,000 species. It's estimated that there are 400,000 to 3 million species out there. So it's not a wonder, really, that these new viruses seem to be emerging and causing having great repercussions in human biology, because it's not necessarily the emergence of new viruses. It's viruses that have been around, and we just don't know about them yet. Even well-studied groups like plants, there are 250,000 described species, and there's an estimate, these tables somehow got screwed up, but they're estimated between 300,000 and 500,000 species of vascular plants. And then there's insects. Insects are just an amazingly diverse group. Over 950,000 described species of insect, and an estimated 8 to 100 million insects. So just incredible amounts of biodiversity out there. And I mean, just the range, 8 to 100 million. That range is so large because we just don't have even good ideas of how to estimate the numbers of species that are out there. That's how much our knowledge is lacking in the area of biodiversity. We're also interested in studying biodiversity in relationship to science and society, and how we as a group of organisms can benefit from our knowledge of biodiversity. So first of all, newly discovered species will multiply the society's inventory of usable resources. New species become very important in agricultural development, in pesticide resistance and transgenics, these sorts of things. The discovery of in pharmaceutical industries, the discovery of new species can be instrumental in developing new medicines and new crops, or improved medicines and improved crops. The new data contributes to the knowledge necessary to sustain and use biodiversity. If we don't understand what's out there in terms of biodiversity, in terms of organismal diversity, it's hard to figure out how to preserve it, isn't it? You need to know what's out there before you can preserve it. And let me tell you, as a conservation biologist, it's an uphill battle trying to do conservation biology when you don't have a good handle on the species diversity in the first place. So gaining that information will allow us to put forth much better efforts in terms of conserving the biodiversity that's out there. Thirdly, knowledge of species diversity will assist in the discovery of new products and the selection of new and improved food crops and medicines. I've touched on this a little bit, and I'll give you an example following this slide, where this knowledge of new species and properties associated with these species become instrumental in developing crops and improving crops and developing new medicines. And then finally, this biodiversity information provides us with baseline data to monitor the global climate and ecosystem change. So I'm sure you've all heard about global climate change and greenhouse effect, and it's very hard to predict what the effects of a warming a warming environment are when you don't know what's out there in the first place. So by doing basic biodiversity inventories, finding out what is in various localities, and then tracking those communities over time, you start to get a handle on how environmental changes can change ecosystems and community structure. So let me give you an example. Here's a paper from Science. This is a frog from Ecuador that was discovered about 20 years ago. When it was first discovered, biochemists started looking at this frog because it secretes a toxin that they isolated and developed, and it turns out that this toxin is an exceptional painkiller. It's on the order of a painkiller like morphine, but without the side effects of addiction. So it's an extremely promising drug that came from this frog that you, first of all, wouldn't have known about if the herpetologist hadn't described the frog in the first place, right? So the herpetologist describes this frog and notes this toxicity in the secretions on the skin. Biochemists pick up on that and start investigating it. The interesting part of this study was that in the article in Science, the investigator, the biochemist, suggested that his work was hampered for over 15 years because this frog was subsequently listed as an endangered species. So he couldn't go out and collect it anymore to get the secretions from the skin of this frog. But if this biochemist knew something about systematic biology, knew something about the interrelationships of organisms and organismal diversity, he could have easily looked at the genus of frogs that this species comes from and looked at sister species and noted that those sister species also have toxicity and could have used one of those that were not endangered, thereby still doing his studies, only he would have got them done 15 years earlier, and then not threatening an endangered species as well, preserving that biodiversity. So it's important to not only catalog these, but make that information accessible and also educate researchers in these other areas about biodiversity and how they can use that biodiversity in their own studies. So let me also try and suggest to you that it's important from a scriptural aspect as well. So this comes from Doctrine and Covenants, section 59, verses 18 through 20. Yea, all things which come of the earth in the season thereof are made for the benefit and the use of man, both to please the eye and to gladden the heart. Yea, for food and for raiment, for taste and for smell, to strengthen the body and to enliven the soul. And it pleaseth God that He hath given all these things unto men. For unto this end were they made to be used, with judgment, not to excess. I think there might be a little more, but for some reason these slides are getting cut off a little bit. But the idea here is one, it's important to know what the species diversity is out there before you can make benefit out of them or to please the eye and gladden the heart. And secondly, to note that these organisms aren't just there for us to use, and certainly not to abuse, right? Not to excess. But these organisms are there for aesthetic reasons as well, to gladden the heart, to please the eye. There are aesthetic reasons scripturally to have these organisms there and to understand how they're related to one another and how they interact as an ecosystem. So I want to talk now about some of the studies that are going on here at BYU and associated with biodiversity. And as you can see, we've got a number of them. And I'm just pulling off a few examples from our zoology department, starting with Jack Seitz, who is doing work in the Amazon, so we will get there. And then Duke Rogers. Jack is a herpetologist working on Amazonian river turtles, conservation biology of this species of big river turtle. Duke Rogers works on small mammals in Mexico, central Mexico principally. Mike Whiting is an entomologist in our department, works on insects, and has recently returned from a trip from Papua New Guinea. All these guys actually work all around the world, and I'm just sort of taking one snippet of what they do to show you today. Riley Nelson, not to get too far away from home, to bring it back home, he's involved in an extensive biodiversity survey in Grand Staircase Escalante National Monument. And this is work that he's doing with Richard Bauman, both of them associated with the museum as well. There's a group from botany doing the vascular plants, Stan Welsh, and then there's also Duke Rogers is doing the mammals, and there are a number of people in zoology involved in this inventory of the national monument in terms of biodiversity. And then finally, I'll wrap up with some of the work that I've been doing in Australia and Madagascar, Chile, and Brazil. So first, here's Jack enjoying a boat ride down the Amazon with an undergraduate and with a colleague from Brazil, undergraduate from BYU. Most of these research experiences, a lot of these trips that we do, we bring undergraduates with us to do collecting. We also employ a lot of undergraduates on campus to deal with the aftermath of collecting. Right? A lot of these trips, depending on the organism, we're either collecting whole organisms and bringing them back, or in this case, these are endangered turtles. It's a highly endangered species that they are collecting tissue samples from. In some of the work that I do on conservation, in conservation biology, where we're working on endangered species of freshwater crayfish, we take just tissue samples in non-destructive sampling. Blood samples are often taken. So a lot of the material we bring back are samples, not organisms, but we do also bring back a number of organisms, and they are all housed at the Bean Museum. So this is the turtle that Jack works on. It's a freshwater river turtle that has a lot of behavior similar to marine turtles. It has natal homing. They go back to the same beach every year to lay eggs. And one of the things Jack's interested in is looking at the genetics of the species and how much gene flow there is up and down rivers, and whether there are distinct populations of turtles in different river drainages in the Amazon. And to do this is a collaborative project with the Brazilian government and some researchers in Brazil, and they go out on their boats and cruise up to beaches and beach their boat and dig for eggs and take samples from the eggs and look at hatchlings. And then they bring this material back to BYU. They do some genetics and look at DNA and can infer from that how whether there's gene flow going on within and among drainages. So it's an important piece of work for the survival of the species to be able to know how they are migrating, how specific beaches are, how mating occurs, who's doing the mating, and that sort of thing. And you can figure this all out with some genetics. The other point is that when you do these sorts of studies, they open up the door for doing more extensive educational experiences. So this is a group that Jack cites, Riley Nelson and Mark Belk from our department. They took a group of undergraduates, a couple of undergraduates from BYU, and coupled them up with 12 undergraduates from Brazil, from 6 different Brazilian institutions, and did a biodiversity experience in Brazil, in the Amazon. And this is the group of them collecting basically anything that moves, figuring out what it is, and then letting it go again. So they had a great time. Secondly, we're going to talk about Duke Rogers. He's doing a lot of research and training in Mexico. And for some reason, Duke gave me a bunch of slides to use from his Mexico trip, but he didn't have any mammals. He's a mammalogist, but he had all these slides of birds and snakes and lizards and things. So I put a couple of those on. But he does principally mammal population genetics and systematics, looking at relationships among species. But the other thing that he's interested in and involved with is Duke has 2 former students, PhD students, who got their PhDs here at BYU, who are now who are Mexican, are back in Mexico in positions there, academic positions, and they are in charge of a biological reserve down there. And this reserve has has associated with it this nice sort of educational complex, has housing, has classrooms, and we are now in the process of establishing an undergraduate student exchange, basically to give some of their students up here to get the experience with the DNA sort of aspects of things, working on our automated sequencer, learning how to analyze sequence data, having experience with our group here, but then also send our undergraduates and some graduate students down to Mexico and experience this amazing country. Mexico is one of the few countries that's considered a mega diverse country. That is, it houses a large percentage of the world's biodiversity in this country. And this reserve happens to be right in the middle of the diverse areas. So lots of opportunity for undergraduate research, lots of opportunity for new discoveries of organisms, and lots of opportunity for fun. The third member of the group here is Mike Whiting. He's like me, been around for about 4 or 5 years here. He's our insect systematist. He's just gotten back from a 3-month trip to Papua New Guinea, where he was supposed to be collecting fleas. That's what he was being paid to collect. But he also works on a bunch of other insects, and it turns out the flea work didn't go real well. But the walking stick work went great. In fact, this blue walking stick apparently is a new species that's never been seen before. There's never been a blue walking stick described in general. Mike would take these. He took one graduate student with him, Allison Whiting, who happened to be his wife, and then a colleague who does flea biology. And they got in these small planes and flew into these remote communities and would pay community members to go collecting for them, and they'd do some collecting, and they'd bring back all this insect, and then Allison does work on skinks, and so they bring back these, and they're sort of like a lizard, so they bring back all these organisms and they'd catalog them. And Mike, again, this sort of research, you bring back these specimens to BYU. They're housed at the museum. The DNA is used to look at relationships among the species, and those are used for looking at the evolutionary history of these organisms. So here's Allison in the midst of dealing with all these skinks that were collected. The way they collect these is kind of interesting. They have this sticky paper that they lay out as a trap, and they lay it out in a sunny area. When the skinks come out to sun themselves and warm themselves, they get stuck to this paper. And you start getting little skinks that are stuck to this paper, and then the big skinks see the little skinks sitting out there, and they want to come eat them. So they come out and they get stuck. So you get this piece of fly paper with 3 or 4 different lizards, starting with small ones and getting bigger and bigger. And they collected some interesting frogs and lots of skinks. And this is Allison's doctoral dissertation work being funded by, to a certain extent, by the National Science Foundation to do this sort of work. While they were there, one of the side benefits of being a biologist and going around the world to collect specimens is you meet people and you meet a diversity of people. You really get into having an opportunity to get into a diversity of cultures. And here, just Papua New Guinea is an amazing little country. Okay, it's right over here, and New Guinea has like 70% of the linguistic diversity in the world is on this island. Just an amazing group of diverse people with diverse languages, diverse dress, diverse cultures. It's really an amazing place to go and do research. This is Riley Nelson. When I asked him for some slides, he claims to have a whole series of bug on my face slides. This is the latest from a collecting trip down in Grand Staircase Escalante. He's looking at various insects and arachnids down there, basically doing an inventory, trying to figure out what is there. When they do this collecting, you set traps for these bugs, and this is what you get. You get a whole mess of stuff when you're doing insect collecting. It's not like the herp collecting where you get a lizard. When you set traps for insects, you get a lot of insects, and And a lot of the work is when you bring these piles back to the museum and you have to start sorting through them. So if any of you undergraduates want to volunteer to sort bugs, there's a lot of work. A lot of work in bug sorting. So we use we use undergraduates to do a lot of the preliminary sorting, because undergraduates can differentiate between a fly and a moth and a cricket pretty well. So you can do the preliminary sorting, and then what happens is then one of the curators has to do some of the finer scale sorting, and then you get to the point where the curators aren't any good, and you have to find the world's experts in that taxonomic group and send them to that person to figure out what species they are or what genus even they are. And a lot of times for those organismal groups, the world expert doesn't exist, or he died 10 years ago is usually the case. So you know, this work is real labor intensive, and it also you run into these problems, these roadblocks in categorizing biodiversity because the expertise just isn't there. So that's one of the things we have to deal with. Again, these collecting trips provide wonderful opportunities for undergraduates to go out, in this case in southern Utah, bask in the sun and collect insects. But they also provide wonderful opportunities for research back on campus once they've done the collecting, to come back and do sorting at the museum, take some of those specimens and do some genetic work in the lab, collect DNA sequences and look at how they're related to one another. So the projects have many facets to them, and undergraduates are involved in every step of the way. So, and then this is just to give you an idea of some of the things I work on. I work on various crayfish species. This is a friend of mine, Pierre from Western Australia. He works at, he's a professor at Edith Cowan University in Western Australia. And crawfishing, there are people in the room who can attest, is not that fun. You basically, if you're a good digger, if you have a big shovel, you'll do all right. It's a lot of digging to get some of these little things out. There are some fun aspects. This is a cave species. This species of crayfish has no eyes, no pigmentation. Caving is a good time. That's fun collecting. The digging, I like to send my postdocs out for that work. All right, so crayfish, it turns out, are also important in various areas in terms of a dietary source, a source of protein. This is a shot from Madagascar. I received funding from the National Geographic Society to travel to Madagascar, Brazil, and Chile to collect freshwater crayfish. In Madagascar, all around this national park that I was collecting in, the livelihood of a lot of the communities was based on freshwater crayfish. And because of the dwindling ecosystem that the crayfish is housed in, the deterioration of it, the pollution of it, crayfish populations are going down. And you couple that with a lot of harvesting, and in some areas over-harvesting of the crayfish, this organism that these people depend on for their livelihood is diminishing. And so we're doing a lot of work now in Madagascar to look at the population genetics of a few of these species, look at how genetically differentiated they are. I'm collaborating with a PhD student at Cambridge University who's looking at how the natives utilize these different species and which are preferred and how many they're harvesting and where they're getting them from. And we're going to try and put all this together into a management plan for the different species that the indigenous people use in Madagascar, so that we can develop better approaches to making this a sustainable resource and not a resource that will be overutilized and then gone. Okay, so that's part of biodiversity studies is making these is developing approaches to keep the species around and in healthy populations so that they can be effectively utilized by indigenous folks. The other interesting thing that really struck home with me in Madagascar, I was there for 2 weeks, was paid by the National Geographic Society to fly there and collect these things. And when I was there, I stayed at Ranamafana National Park. And at the park, they have, I don't know if you've heard of this program called Earthwatch, but it's a program where people pay a lot of money to hook up with scientists and go off and do research. And there was a group of these earth watchers there when I was there, working with one of the professors from SUNY at Stony Brook who works on lemurs. And these folks had all paid $4,000 to $5,000 each to come there on their vacation time, right? And that didn't include the flight of the cost, or the cost of the flight, which was impressive. So it really struck home that as a biologist, it's fun to know that I'm getting paid to do what other people are paying lots of money to do for their vacation, right? So let me just say, biology in general is a blast. And if any of you are even thinking about it, you ought to explore it, because it's a wonderful job to be in. All right, so there are additional benefits to these biodiversity studies. First of all, these methods that we're developing to estimate phylogenetic relationships, basically gene genealogies, looking at how different organisms and different genes are associated with one another, are evolutionarily linked to one another. These methods are being used in a number of other areas. They're being used to look at the evolution of infectious disease, the dynamics of infectious disease. They're being used to study molecular biology, to predict function of unknown genes. They are a very powerful tool to subsequently do comparative frameworks, to look at ecological differences, to understand the evolution of different ecologies and things like that. So, and a lot of this is very computer and mathematics intensive work. So we have ongoing collaborations with the statistics department, with people in the computer science department, to develop better approaches to estimate these genealogical relationships among genes. And that's an exciting piece of work. And we also apply these techniques to other areas, one of which that I apply it to is the evolution of infectious disease. In particular, I've been heavily involved with looking at the evolution of HIV. And so we can use these same methods that we use to classify organismal diversity, classify biodiversity, look at species relationships, use the same methods to look at the genetic diversity of HIV across the world. And you use that to study the epidemiology of HIV, to look to guide you in drug development, to guide you in vaccine development. This is a critical step, understanding the diversity of HIV and how it's distributed geographically to do any effective treatments for HIV. And my lab group's been pretty heavily involved in looking at drug resistance and now in vaccine development as well. Secondly, it's this as an example of using this biodiversity as a framework for other studies. Here we're using it as a framework for conservation. As I alluded to earlier, I'm personally not happy with just cataloging all the biodiversity. I'd like to keep some of it around. And just to point out the issues with crayfish, there was a study recently done in 1997 by the Nature Conservancy that looked at the number of species in various groups and compared them to the number of species that were considered endangered to some degree. And crayfish has a dubious distinction of being one of the two groups, freshwater mussels being the other, in North America, with more species endangered than are safe. So conservation biology, especially in terms of freshwater crayfish, as a representative of that freshwater ecosystem, and you can see it's that freshwater ecosystem that's really in trouble with the bottom three here, freshwater mussels, crayfishes, and amphibians, and then freshwater fish. All these are in the same habitat, that freshwater ecosystem, and that one is one that's really getting torn apart. So we use this biodiversity information to look at conservation. So one of the first things we do, one of the things we do a lot of in my lab, is sequence DNA. So we get a lot of G's, A's, T's, and C's, and then we try and make heads or tails of it. And we do that by looking at evolutionary relationships, reconstructing what's called a phylogeny, and a representation of how different species are related to one another. And then we can look at these species and we can ask, where are they geographically? And we can use that correlation of the genetic diversity, the amount of diversity in the phylogeny, coupled with where they are geographically, to look at conservation priorities. And we've done that in Australia. Australia has been divided up into a number of different bioregions, and these different colors show the different bioregions. So what we've done is reconstructed the evolutionary history of a bunch of crayfish from Australia, and then used that phylogenetic information coupled with species counts, the raw number of species diversity in these different areas, to rank these areas for conservation priority. So this gives some data to politicians, to conservation managers, to make decisions based on real data. And what we found is that this, for freshwater crayfish anyway, this area in southern Victoria, Tasmania, southwestern Western Australia, and then southeast Queensland were the areas of real high diversity and ranked high in terms of conservation priorities. So let me just summarize then that there are plenty of biodiversity studies going on here at BYU. They are international in scope. We go all around the world. And we don't just go around the world and collect stuff and bring it back, but we establish collaborations with international colleagues. And we keep close ties with those colleagues and bring them here a lot for visiting research, as well as go to their institutions, give guest lectures, exchange students, and we're going to be doing more and more of this in the future. And finally, as I've alluded to a number of times, there are excellent opportunities for undergraduates to experience biodiversity studies, to get out in the field and have some fun, collect some organisms. Perhaps you too could have an orthopteran on your face for a nice photo. And then finally, just to give you a little flavor, the international group that we have working at BYU right now in the zoology department, just in our little group that does systematics in the biodiversity studies, we have graduate students from Bolivia, Chile, Spain, Mexico. We have postdoctoral fellows from Australia, England, Brazil, Spain, Mexico. And we've had visiting scientists from Mexico, Germany, France, Argentina, Brazil, Spain. I'm sure I'm leaving out a lot. And we do have a couple of Americans too. So I won't leave them out. And we've had generous funding from a variety of sources to do these biodiversity studies, to employ these undergraduates, to bring them with us into the field, and the graduate students as well. We've been generously funded by the National Science Foundation, Bureau of Land Management, Division of Wildlife Resources here in Utah, the Alfred P. Sloan Foundation, National Geographic Society. We've been well funded by the university. The Vice President's office has given us, they have an emphasis on undergraduate mentoring research opportunities. We've been given a number of, a good amount of money from the Vice President's office to support undergraduates doing this research. Likewise, we've received some matching funds from the college, from our dean, for undergraduate research experiences. The Office of Research and Creative Activities has provided a number of scholarships for our students. They've been very helpful. The Office of Graduate Studies has provided us with funds to send graduate students to some of these different countries and different areas to look at biodiversity. And then obviously, the reason I'm here is we've been funded from the Kennedy Center as well, and we hope to continue that rich tradition. All right. So, to conclude then, a quote from Teddy Roosevelt, a message to Congress on December 3rd, 1907. To waste, to destroy our natural resources will result in undermining in the days of our children the very prosperity which we ought by right to hand down to them, amplified and developed. Thanks for your attention.
We have time for just a few questions before we need to break, so let me open up. Let me ask the first one, Dr. Crandall. You talked about the need to continue inventory and cataloging. Am I right in assuming that there's a relationship between development and inventory? That is, in developed countries like the United States, the inventory would be more progressed, and less developed countries would be less progressed?
Well, there's actually an interesting relationship there in that the inventorying is well progressed in developing countries, but most of the biodiversity is in less developed countries. So most of the need for inventory are in these less developed countries. So no, it's more a latitudinal gradient. So when you get closer to the equator, you have more diversity on average than further away. And developed countries just happen to be further away from the equator. So.
Questions from the audience? If not, then thank you for being here.
right. Appreciate your attention.